I know I was always very curious, always asking why questions, and I was very interested in how ice formed. I thought it was phenomenal that you could put water into the freezer and then later on you could bring out ice cubes. And so Mom and Dad caught me with the freezer door open, staring at the ice tray. They were very understanding, and they helped me set up my first experiment, which basically was to have an ice cube tray in the house, and then since we lived in a colder climate, we could put water in an ice cube tray out on the porch. It was very exciting, my first controlled experiment. My name is Amelia Ahern-Rindell, and I am a scientist and a college professor. Curiosity is something that slowly gets weeded out of kids. I mean, as I was growing up, I was constantly asking why. And after a while, you kind of are discouraged, I think, from asking that why. So my interest in genetics happened relatively early. I used to babysit, as many young people did back in the late 60s, early 70s. And I happened to babysit for a family that had 3 young daughters. All had the autosomal recessive condition known as cystic fibrosis. And I think what struck me mostly was the fact that here were 3 young girls who, through no fault of their own, had this condition. And as a result of it, their lives were not only shortened, but definitely the quality of their life was impaired. And so I wanted very much to answer that question of why do they have this condition and what can we do about it. This kind of inspired me to move into the realm of undergraduate research, into doing research and going to graduate school. For a while there, I thought, I would like to become a doctor and help individuals like this. But as I had more exposure to different opportunities to engage in research, I realized that I actually really enjoyed that more than I thought I would working with patients. Plus, I think I had a sense that I was somewhat of a softie and that I would probably have a hard time kind of separating myself from my patients. One of the things I did as an undergraduate student was a thesis on some research pertaining to what are called the lysosomal storage disorders. And within the cell is a small, kind of a vacuole that we call an organelle. And that organelle is kind of the recycling center of the cell. We call it a lysosome. And it's full of various proteins that help break down macromolecules in the body. And that would be things such as proteins, carbohydrates, fats, etc. So these lysosomal storage disorders result when one of these proteins don't work properly. And as a result, the material it's supposed to break down accumulates. And that can lead to all sorts of problems for the cell and then on a system-wide basis. One of the conditions that I have worked on for a very long time is called GM1 gangliosidosis. We call it GM1 for short. But in essence, GM1 is a major component of the membrane, kind of the layer surrounding a cell. And if you can't break that down appropriately, it will accumulate and it leads to a lot of problems, particularly in the nervous system. So neurons, the major cell type in the nervous system, degenerate. And currently this is a fatal condition. There is no real treatment. There is no cure. And so I've always been interested in trying to figure out kind of the mechanism, what's happening at the molecular level. I think what was most inspirational about my experience as a postdoc was having parents of patients with GM1 call the lab and ask how we were doing, how we were progressing. They knew we were working on this, but obviously we were many, many years out. And and to listen to their anxiety, their desire to help their child in some way, also made a big impression on me. And so that's kind of a pivotal point where I decided I didn't want to just do research. I didn't want to go into medicine. I wanted to become a college professor where I could not only teach, but I could continue to do research. And that, in part, is why I ended up here, because I can do that. I can kind of have it all. I remember when I was an undergrad, and a lot of the learning I did was based on the professor standing up and lecturing at you. It was kind of the idea that I'm just gonna, toss my knowledge out there and hopefully you'll pick something up. And when I became a teacher, I wanted very much to engage my students and have them actively involved in the classroom. So very early on in my career, I was fortunate to get some National Science Foundation funding so that I could teach a lab that brought research directly into the classroom. And over the years, 20-something years now, I've kind of continued to do that. And when I teach my genetics lab, what the students are working on is not these cookbook typical experiments with fruit flies or your old-fashioned type of experiments. They're helping me with my research. And what's really fun about it is I don't know the answer. And so rather than them coming in and saying, this is what I'm supposed to get, if I didn't get this, what did I do wrong? It's more of an affirming type of experience where the students are learning by doing. And so, they're in little groups and they each get a portion of the sheep beta-gal gene to help clone. And they are convinced that they're going to find in their affected sheep the mutation. We kind of, during the first semester, go through some of these techniques. So they're learning all the techniques, and in essence, they're kind of repeating work that I did. And then the second part of the semester, we get right into it, and they start using the sheep tissue. And they isolate the DNA from a normal and an affected sheep. And then they get their little exon. They have to design primers to amplify the particular gene area we're interested in. And eventually, they produce a poster. And we have a poster symposium at the end of the semester. And I invite administrators, faculty, and they come and they see the students present. And what I think is special about this is that within the confines of a classroom, the students are actively learning. It's hands-on, it's authentic. They don't know what the answer is until, hopefully, they discover it, they identify it. But I think it makes the science fun. It makes it interesting. It gets back at being curious and being able to ask questions and figure out why, rather than being told a whole bunch of facts that, in one ear, out the other. They get a chance to really think like a scientist. I'm afraid we're turning students off to knowledge, and knowledge really is power, not because in and of itself it gives you power, but having information and being able to use it in an applicable way to solve problems. That is power. See, you learned something.